In this video, we're going to take a look at the endospore stain, and we'll need a couple of supplies as usual. We need our glass slides, of course, Sharpie to make some labels, your slide retention device, your cultures, your loop, and today we're going to be using a boiling water bath along with our malachite green solution and our safranin stain right here. And the point to the water bath is to basically open up the spore coating so that our malachite green dye can get in there. And then when we go through this process of the endospore stain, we're going to decolorize, and the malachite green color will come out easily from the body of the cells, but it'll be trapped in the spore coating. And again, because we're decolorizing our cells, we're going to counter stain with our basic diagrams safranin stain here so that the body of the cell will come out pink, as we'll see hopefully later on. To start this process, we'll do like we've done in our previous lab with the direct stain, and the green is from cleaning out the beaker there that I got. But we're just going to take a loopful of our water and come in here and make our smears in that loopful of water. So I have an unknown soil, and I'm just going to cool this off by stabbing the side of the plate where there's no cells there, and I'll get a little bit of one of these colonies off of here. And again, you don't need very much, just touch it to the edge of the colony really there, and you'll get some of the newest cells. And I'm going to just put this in the middle one there because that's my unknown. And then we will put two other bacteria onto this slide. And the one thing I will mention here is just because you are using a bacteria for an endospore stain does not mean they make endospores. Endospores are something that are made when the cells are in an adverse environment where they can go dormant and protect their DNA. So we don't always get spores. We have a few types of bacteria that we use that are more reliable at making the spores. So just go ahead and go through that process and make your smear with your various cultures, and in this case our unknown culture. So here's my last one, just a reminder, aseptic technique, sterile loop, flame the lip of your glass, flame it again, put the lid back on, and then we can go in and finish making our smear with the last of those cells. And so you can see the smear that we have here, just light. The one that I pulled off the plate, you can see a little chunk of that right there. But we're just going to let this sit for a few minutes so that the cells will have a chance to air dry.